Hello, my name is Amelia Cosgrove,
Hi, I'm Sydney Sanchez.
and our project is on the Casc gene. So the Cas gene is a protein coding gene, which codes for the Cas protein, which is a member of the membrane-associated guanylate kinase protein family, or the MAG-UK family. And the Cas protein is an important gene to study as it is important for neurodevelopment, as it functions as both a gene expression regulator as well as a protein function regulator during neurodevelopment. It is also important as a scaffold protein and signaling of neurons to the control of ion movement. The Cas protein is most abundant in brain tissue. However, it is also present throughout the entire mammalian nervous system. The Cas gene is important to study as it is associated with multiple disorders such as the X-linked intellectual disability, microcephaly with pontine and cerebral hypoplasia, or MICPCH, which is the one we're going to focus on for this project. So for this project, we are suggesting a multi-omics proposed methods design using transcriptomics, epigenomics, proteomics, and phenomics. So as I mentioned, we're going to be focusing on MICPCH, postmortem brain tissue samples, as it is the most severe CASC-associated disorder and is the most common one in females. This disorder is believed to be a loss of function mutation and typically causes death in early infancy for those affected. Through comparison of the MICPCH postmortem brain tissue samples to age-matched neurotypical postmortem brain tissues, we hope to increase our understanding of the mechanisms and pathways that lead to these CASC-related disorders, specifically through the analysis of regions of the brain such as the cerebellum, pons, frontal lobe, and amygdala. So in a preliminary study conducted in a Cas knockout cortical excitatory induced neuron stem cells, a complete Cas loss of function was introduced to these neurons and a differential gene analysis with DEC2 was performed. And through comparison of these two Cas null cell lines and wild type, they were able to identify 876 DEGs in immature neurons. And DEGs are differentially expressed genes. And through this analysis, they were able to identify 420 upregulated DEGs and 456 downregulated DEGs. And then through unbiased gene set enrichment analysis, they were able to relate these DEGs to specific pathways for cell adhesion, cell projection and morphogenesis, neuronal development, and synaptic membrane and structure. The same analysis was then also performed in mature neurons, and through the differential gene expression analysis, they identified 1,743 DEGs. 905 of these were upregulated, and 838 of these were downregulated. Then, through unbiased gene set enrichment analysis, 422 significantly enriched pathways were identified, such as pathways for synapse assembly, cell junction adhesion, axon development, neuronal apoptosis, cell projection morphogenesis, neuronal development, and additional cellular components. For this project, we seek to build off of these preliminary analysis through additional RNA sequencing with poly-A tail enrichment, so quantifying the mRNA abundance in those multiple brain tissue samples that were previously identified, and then perform differential gene expression analysis based on the mRNA that is sequenced. Through this, we hope to identify the DEGs in multiple different brain tissue, as well as confirm those previously identified DEGs in the neurons. And then lastly, we hope to detect novel DEGs in the brain tissue samples that will be analyzed for this project. And through this analysis, we hope to identify pathways that have been affected by the Cas dysfunction.
For the epigenomics portion of this study, we did do some preliminary analysis. We were hypothesizing that we would find changes across chromatin accessibility, histone marks, and DNA methylation in areas that have been linked to neurodevelopmental issues. We ran ATAC-seq and got some differential accessible peaks and distal enhancers that were linked to synaptic genes that, for example, included RELN. We also ran the cut and run software, which reduced that acetylation to 45 enhancers in the tissue. And then we ran WGBS which then found hypermethylated DMRs at those downregulated genes. And then finally, we did kind of a connection with the transcriptomics by running an RNA-seq analysis that found about 400 DEGs that were also linked to downregulation of RELN and those synaptic scaffolding and cortical layering genes. This helped us find that Cas does maintain active enhancer states necessary for the neurodevelopmental gene expression and how loss of Cas does disrupt these regulatory programs as well. This is part of the figures that we concluded with our preliminary analysis, and what you see here on the left is a genome browser that compares the RELN region to a control and an MIC-PCH sample tissue. We did find here that distal enhancers upstream of the RELN show those reduced ATAC-seq accessibility, and then for enhancer activity, we did show or see loss of active enhancer marks for our tissue, and then our methylation track does show that MICPH or MICPCH had increased methylation, which is consistent with repression, epigenetic repression. And then lastly, on our last track here for RNA-seq coverage, we do see that there is less coverage for our tissue as well. And if we were to further explore our epigenomics analysis, we would increase our sample size, as here we only have one control and one MIC-PCH tissue, and then also apply Hi-C to map those Cas-dependent enhancer promoters as well. And then some further proposed methods for a proteomic analysis would include assessing how the Cas deficiency does alter protein abundance and those interactions that we're looking for. And we do know that Cas does have hydrophobic properties, making it more difficult to run this analysis, but we will use a membrane protein optimized method linked with bottom-up proteomics to try to get the best results for this analysis. Then MaxQuant will be used to process the mass spec data analysis that we receive after the proteomic step and try to detect Cas isoform specific fragments. Then we can use a tool called Perseus for differential protein expression between our samples and then identify those downstream changes in the scaffolding proteins like RELN or other signaling pathways to combine and find those RNA protein mismatches as well that could reflect Cas loss.
The last method that we will be using is phenomics. So through a systematic study of observable traits, which will be collected through the request of electronic health records from multiple medical facilities across the United States. We will then analyze this database of clinical phenotypic data for MIC-PCH patients, and we will look for expected phenotype traits which are associated with Cas dysfunction, such as microcephaly, epilepsy, intellectual disability, hypotonia, and hypertonia, as well as hearing and vision loss. Aside from the expected traits, we will also analyze this data to see if there are any other observable traits that might be linked to the Cas dysfunction. And then lastly, we will perform Fawaz analysis of the observed traits and known Cas variants in order to try and identify causal links in the MIC-PCH patients between these traits and the Cas variants.
Some limitations and challenges that we will come across is the cell-specific function expression, mostly how we handle our cell and tissue types during sample processing. And again, we do know that Cas is hydrophobic, so making it difficult to perform some of the proteomic analysis that we have provided. And getting the access to the electronic health records will be take a lot of time because we're trying to access the correct data and make sure we have enough of it. And then just knowing the complexity of running this type of project with the pleiotropic nature of Cas will add a lot of time to our progress in this project. And then to just wrap up, we do just want to quickly show how we did go off of previous transcriptomic studies that did find dysregulation and loss, Cas loss, and then how our epigenomic study kind of followed that up with those histone marks and DNA methylation and MIC-PCH, and then how we can combine proteomic and phenomic analysis to connect those disruptions to protein and phenotypic traits, and then how overall this multi-opic approach is the correct way to map how Cas variants alter neurodevelopment and guide future diagnostics for therapeutic targets and other Cas-related disorders as well. Thank you, and these are references. We have many more, but they are found in our grant proposal. Thank you.